Hi everyone, welcome to the third lecture of the RNA algorithms course. Today's lecture is RNA structure 101. We will cover the basic terms and concepts of RNA structural studies on different levels. So we will start with RNA sequence, which is like structure of 1D dimensions. Then we will talk about residue reactivities from chemical probing, which I named like 1.5D because it's somewhere in between of sequence and secondary structure. Then we will talk about RNA secondary structure, some additional interactions on top of secondary structure, including tertiary base pairs and coaxial stacking, which is sometimes named as 2.5D. Then we will move to 3D structure and then some intermolecular interactions of RNA with other molecules. So we will start with RNA sequence. So RNA sequence is a string in 4-letter alphabet. So in computers we have it just like here. So we have a start, we have the first nucleotide, we have the last nucleotide. This is how it looks like on the chemical composition. So these are the 4 monomers that we have, A, G, C, and U. Each monomer consists of a base of one of 4 types. A sugar, which is a ribose in case of RNA and deoxyribose in case of DNA, and a phosphate group of phosphorus and 4 oxygens. The start of the molecule, and yeah, it is a linear polymer, so we have a start and an end, and we don't have any branches. So it's just a single long linear string. We have 5' end, which is the name of the beginning of the molecule, and 3' end is the name of the end of the molecule. And this is how a 3D structure of transfer RNA or a tRNA looks like. This 3D structure is in rainbow coloring scheme, one of the popular coloring schemes for visualization of 3D structures. It starts with the 5' end in blue and ends with the 3' end in red, and all in between are rainbow colors in the order of the rainbow color. So we start with blue, go through light blue, green, yellow, orange, and end with red. So why it is 5' end and 3' end? So we need to first look at the nomenclature of atom positions of the nucleotides. So each monomer, as I said, includes a base, a ribose, a pentose sugar ribose in case of RNA, and the phosphate group. Atoms of bases are numbered with normal, regular Arabic numbers. So these are like 5 possible residues, including thymine for DNA. So here we have pyrimidines, which is a single ring base, and purines here are double ring bases. Here the atoms are numbered counterclockwise. Here atoms numbered clockwise order. On these diagrams, we have carbon atoms just omitted from the picture. So this is a carbon atom, this is a carbon atom, this is a carbon atom. Then nitrogens are shown with N, oxygens are shown with O, and so on. This is the glycosidic bond between nitrogen atom of a base and the carbon atom of a sugar. And to distinguish atoms of a ribose from atoms of a base, atoms of ribose numbered with additional prime symbols. So here we have carbon C1 prime, C2 prime, this is oxygen O2 prime, carbon C3 prime, O3 prime, C4 prime, O4 prime, C5 prime, and this is O5 prime. So these are the numeration, this is the numeration. And the O5 prime atom is the place of where the phosphate group is connected, and another position of the following phosphate group is O3 prime atom. And when we look at the backbone of an RNA, so we have like ribose, phosphate, we always start from 5' side. So when we have these 2 riboses, this will be the first ribose, and this will be the second ribose. So this is the 5' end because we start from 5' atom of a ribose, and this will be the 3' end. So the phosphate connected to 3' oxygen is like the following one. right? So this is why the beginning and the end are called the 5' end and 3' end. And this is how a helix, how an RNA helix may look like in 3D. And this is actually the second popular coloring scheme of RNA visualizations. Here we have coloring based on base types. So here, like, all the carbon atoms are colored based on the base. So adenine carbons colored with red, guanosine carbons colored with green, uracil in blue and in light blue, and cytosine in yellow. But all the non-carbon atoms are colored with their colors. So oxygens are colored in red, nitrogens are in blue, phosphorus in orange. So this is how normally we look at the RNA, an RNA in 3D. Moving forward, so each base, each nucleotide actually, yeah, and here I should have mentioned here that we have a nucleobase which includes only a base. If a base is connected to a ribose with no phosphate, then this two-component monomer will be named nucleoside. And if we have a phosphate, a ribose, and a base, that's called a nucleotide or a nucleoside monophosphate. So nucleoside monophosphate is a nucleotide. And so each nucleoside or nucleotide may have a range of different conformations. One of the most famous or most frequently mentioned conformation is either anti or syn conformation of a nucleobase relative to the sugar. So this is an anti-conformation where the base is located relative to the ribose in this way, and if we flip the base, then we will end up with a syn conformation. Anti-conformation is a standard conformation of A-form helix of RNA, and in general it's more than 90%, maybe more than, well, roughly 90%, slightly more, of all nucleotides in RNA structures are of anti-conformation, and around 10%, less than 10%, are in syn conformation. Another popular conformation frequently mentioned is a sugar packer of a ribose. So ribose is a 5-atom plane, and sometimes not all 5 atoms lie in the single plane. So we may have conformations when 1 or 2 atoms lie outside of the plane, and frequently it is either C2' or C3'. So this is an example of C2', 2' endo conformation where C2' is above the plane. This is an example of C3 endo conformation where C3' atom is above the plane. If it would be below the plane, it would be C3' exo conformation. And likewise here, if C2' would be here, it would be C2' exo conformation. Or we can have both atoms lying outside the plane, normally in different orientations. So this is an example of C3' endo, 2' exo conformation. So we have 2' carbon below the plane and 3' carbon above the plane, and here is the opposite conformation. This is an example of the most frequent conformation of RNA nucleotides, which is 3' endo conformation. So this conformation is, this nucleotide shows an example of this 3' endo conformation. Now, all these conformations, like anti-syn or 3' endo, 3' exo, 2' endo, are defined by specific values of torsion angles. And here you can see all the different torsion angles that we can measure for RNA nucleotides. And mostly we designate them as Greek letters. And a torsion angle, what a torsion angle is? A torsion angle is defined by 2 atoms and 3 covalent bonds. So for example here, let's say this angle theta is defined by these 2 atoms, C4' and this phosphorus, and 2 covalent bonds of theirs, which are not between them. So this covalent bond and this covalent bond relative to this axis defines the angle theta. So it's an angle between this and this covalent bond. And actually, we have Greek letters for actual torsion angles defined by real covalent bonds, and this theta and eta actually are pseudo torsion angles because they are defined by pseudo covalent bonds, because C4 prime and phosphorus are not connected. They have like C5 prime and oxygen between them. So in coarse-grained representations, people use pseudo torsion angles defined by like between coarse-grained representations. So like this is a reduced representation of a backbone, and these two pseudo torsion angles define the conformation of this backbone. If we have the full atom model, then we have much more torsion angles, as you can see. So normally for phosphorus-related angles, we have alpha, beta, gamma, and so on. For ribose-related angles, we have numbered nu letters. And for glycosidic bond, we have chi angle. So actually, chi angle is the angle that defines the anti-syn conformations. And for some intermediate chi values, we can have some like Gaussian orientations, but these are rarely mentioned because those are intermediates. Now, apart from 4 standard nucleotide letters, ACGU, we actually have a EUPAC, IUPAC nomenclature of different like ambiguous positions. So in some cases, we don't know exactly what the position, what a nucleotide in a specific position is, but we can like narrow down the search from an arbitrary nucleotide. So if it is, for example, we know that the position is placed by purine or A or G, we designate it as an R. So R stands for purine. And opposite, like if it is pyrimidine, we designate it as a Y. If we don't know at all what the position is, completely unknown, then we designate it with N, which stands for any base or nucleotide, or actually unknown. And if it is a gap position in an alignment, we can usually show it with dashes, tildes, or dots. Sorry. Sorry for that. Yeah, and so these are like ambiguous letters for that can have 2 separate values. These are letters for 3 separate values. And in general, this is IUPAC nomenclature. Now, apart from standard bases or ambiguous positions, we can have modified residues. So modified residues are like non-standard residues where we have some modifications of standard residues. So these are some examples. This is like an adenosine with additional methyl group on N6 position. This is M5C, which has an additional methyl group in C5 prime and C5 position, sorry, and so on and so forth. This is pseudouridine. This is actually an example where the uracil base is flipped, like mirrored relative to the ribose, relative to the glycosidic bonds. So normally in uracil we have this nitrogen here, but this is a flipped example. This is, I believe, a mistake. So here we should have the nitrogen. Oh, this is a normal uridine. It's listed in the among the modifications because we have enzymes that add additional uridines at the end of A tail of some mRNAs. So normally messenger RNAs are often modified, but among structured RNAs, ribosomal RNAs and transfer RNAs are the most modified non-coding RNAs. In general, we have more than 100 known natural different modifications. So for a small amount, we have single letter representations. So for example, for pseudouridine, we have a letter P. For inosine, we have a letter I. But for many of those, we don't have single letter, at least common single letter representations. One of the most famous database of RNA modification is Medomics, and also it's worth mentioning the RNA, the recently established RNA, human RNA project, which seeks to identify all the positions of RNA modifications of the human genome. But we will not cover the RNA modifications in this course, more or less at all. Now, how we store sequence data? We have basically for pure sequence data for single sequences, we have basically just single FASTA format. This is how the format looks like. So we have a header line, and then we have sequence lines. header line always starts with a greater sign, followed by the name of the entry. And then we have multi-line, potentially multi-line specification of the sequence. So this is the entire sequence split into fragments of lines of the length 70 in the canonical FASTA format, but in the current usage of the FASTA format, the line lengths may be arbitrary. Now, apart from single sequences, we have a couple of alignment specific formats. One of them is cluster format. So this is how cluster format looks like. So it stores like aligned sequences and may have additional lines specifying conserved positions in this example by asterisks. So these are the names, these are the sequences, and they are split in several fragments. And these are the indices of the end position in each line. Cluster format was introduced by the developers of the cluster aligner tool in 80s, somewhere in 80s. Then the developers of PFAM database of protein domains developed Stockholm format for alignment, which is basically similar to cluster. We also have headers and sequences potentially split into fragments, but also we have specific header metadata section, and we have like consensus or structure section for the entire alignment. So in this example of aligned proteins, we have secondary structure as this line. For RNA, like here would be a dot bracket notation of a secondary structure or a consensus sequence of the entire alignment. So these are like fancy alignment formats, but also you can store alignment data in aligned FASTA format, which is basically the FASTA format where the sequences are of the same length and include gaps. I think FASTA is the most frequently used format. Now, sequence editors. So like the editors where you can look at and edit or rearrange your sequences. There are some specific tools for that, like this Eugene tool, for example, which I think may be just like Windows only. Maybe it's not suitable with Linux, but I'm not sure here. Or you can just use a simple notepad on any system. So this is like a screenshot of a notepad which I use on my Ubuntu. So it's sufficient. Like the only feature that I need is, which I have in my notepad text editor in Ubuntu, is when I have a prompt on a bracket designating some base pair, I have highlighted the partner of this bracket. So this is very important feature that I constantly use. So I don't know of any like commonly used, the most popular sequence editors. So this is a matter of taste. Now the databases of sequences. We have NCBI nucleotide database, which actually stores more or less all the sequences that people, like the humanity knows. One possibility to search for the sequences, like among the sequences, is a BLAST and search online one. So BLAST is a search tool for like homology search tool. On this website, you just put your query sequence and search against like the core nucleotide database or some variants of it, or some like specific subdatabases. So basically, like this is the place to look at when you search for any sequences. Specifically talking about RNA, we have RNAcentral, which is like an aggregation of many expert databases. So this is the list of all the databases that it has. And it basically stores all the sequences that are known as transcribed RNA sequences. So for example, it has non-coding RNAs from Rfam. It has like microRNAs from MIR base, 5S ribosomal RNAs from this database, and so on. So like many expert databases. So RNA specific database, sorry. And unlike this, like NCBI nucleotide includes all the DNA sequences. And of course, the format is interchangeable, so you can just, if you put the RNA sequence into BLAST search, it will search you for DNA sequences as well. So it always automatically transforms U into T and vice versa. But if you need specifically RNA sequences, RNAcentral is the place to go. Now, moving on to 1.5D. This is like some information about secondary structure, which is not yet secondary structure. And this is about residue activities from chemical probing data and what it is. So the idea is as follows. So once we have an RNA which has some structure, we can apply some reagent to these molecules, which modifies specific positions and with different probabilities. So like the more flexible the position is, normally people assume that that the base paired residues are being modified on a lower rate than single-stranded nucleotides. So these are like the spectra of different reagents used in different protocols. So we have one of the most popular shape protocol, which modifies O2' atoms. And of course, like both base paired and unpaired residues have available these O2' atoms, but shape works mostly on the most flexible residues. So the more flexible residue is, the higher modification rate it will have. And unpaired residues are, on average, more flexible than paired residues. Another popular reagent is DMS. DMS modifies Watson-Crick edge of cytosines, predominantly cytosines and adenosines. And here, like, it's a more direct relation toward distinguishing paired versus unpaired, because this atom of cytosines and adenosines are actually not available, not accessible to DMS for base paired positions. So we have different positions which predominantly modify different reagents that predominantly modify in different positions nucleotides that are unpaired. And once we modify these molecules, we then either have a protocol where so we apply reverse transcription to these molecules to get DNA reads from our RNA molecules, and these modified positions are either make the transcriptase fall off the RNA, so the read will stop in the modified position, or to mutate, to introduce errors in these modified positions, which will translate to mutations in the DNA reads. And then we just read this signal, either the stop, transcription stop signal or the mutation rate signal, and we convert all this via Illumina sequences, NGS sequencing, or any other sequencing currently used, into a spectra of reactivities. So the more flexible residue was, the more frequently it was modified, and the higher the peak, the signal peak will be from the chemical probing experiment. So the experiment is called chemical probing, and the overall result is residue reactivity profile for the entire sequence of an RNA of interest. So we can have it for a single RNA, it will be in vitro probing, or we can have this probing for all the RNAs in a cell, so transcriptome-wide probing. We can have a variety of protocols. This is an example of how raw data may look like. So we have actually a plot of peaks in different positions, and here we have the area of, we always have control data for untreated, unmodified experiment, where we don't add the reagent and measure the same thing. So the background peaks. So this is the area of the treated experiment for pair position. This is an area under peak for background. This is the difference, and these are some normalized somehow differences. So ultimately, this raw data translates to normalized reactivities, which are basically normally values from 0 to 1, with some outliers higher than 1. And in some different versions, we have normalized reactivities from 0 to 1, and some versions we have from 0 to 2. I believe now from 0 to 1 is the most common way to normalize reactivities. For positions with missing values, we normally show some negative values to specify missing positions. Mostly it's minus 999 by convention. These are some examples of plots for residue activity plots. This is one of the common coloring schemes. So we have reactive bases in red, mid-reactive residues in yellow and non-reactive residues in black. So non-reactive residues are thought to be base paired. Reactive residues are more likely unpaired. Intermediate reactivity are more or less like unknown positions. And tools that predict secondary structure know how to use these residue activities to guide the predictions, to like modify the predictions and take into consideration these reactivities. And these are some examples of secondary structures predicted with these reactivities. So you can see that mostly like paired positions correspond to black dots, and unpaired positions correspond to red and yellow. This is like by design. So I don't know why exactly this coloring scheme was adopted. So for me it's a bit counterintuitive, because I expect when I see red and yellow, it's like I expect heat-based idea, but when it is heat-based, then yellow should be higher than red. So this coloring, I believe, more like a street light idea, where we have like red light screaming to be like unpaired, and yellow light is to take into account, right? Okay, so we may have outliers higher than 1. Some tools like clip the values higher than 1 to 1. So now more or less it is a common way to clip all the values from 0 to 1 and consider 0 as base paired positions, 1 and as unpaired positions, reactive ones. Now we have databases of different experiments publicly available from different like studies. One of them is RMDB database. It has like many in vitro probing experiments for like up to around more than 4 million sequences. Another example is RASP20, which is mostly stores transcriptome-wide experiments, data of public transcriptome-wide experiments. So also like not a lot, but when you multiply it for how many sequences we have in a single transcriptome-wide experiment, it also will be, I believe, millions. And we have one popular data set of RNA probing from a recent, well, not so recent anymore, like from 2024. There was a Kaggle contest called Rybonanza, where people were trying to predict computationally these residue activities for single sequence input. So, and for that, the dataset was prepared with residue activities either with shape probing or DMS probing for 2 million sequences. And now it is still available, so it is a good dataset for training and testing your algorithms for residue reactivity prediction if you want to develop one. Now, moving on to secondary structure. Now it is full 2D, full secondary structure. So residue activities was not entirely 2D, because you get the information for a single residue, whether it's paired or unpaired, but most likely paired or unpaired, but you don't know the partner of the potential base pair. So now we talk about the full secondary structure and what the secondary structure is. On top of a sequence of an RNA, so this is our RNA, this is its sequence, and RNA also forms base pairs, kind of like DNA does. So you should know that like DNA is a double helix, like a regular double helix, and in DNA we have regular CG and AT base pairs, which do look like this more or less. So this is CG and UA base pairs from RNA. But here the geometry of the base pairs, of the canonical Watson-Crick base pairs, are the same as this in RNA. But unlike in DNA, where we have 2 strands which form a regular double helix, in RNA we normally have a single strand, and some fragments of its single strand form double helical fragments with each other. So like complementary fragments form base pairs with their complementary counterparts. So complementary strand is the strand where you have like complementary partners for base pairs. So like for a strand GUACU, we have a complementary strand AGUAAC, because like all the pairs can form canonical base pairs. So this is what the complementary strand looks like, and these formations of consecutive base pairs form anti-parallel fragments. So if like this, so if we believe that this is 5' end, so this strand goes in this direction, like top to bottom, then this strand goes in this direction, like bottom to top. So the formation of the double helical fragments are always anti-parallel. And for among secondary structure forming base pairs, we consider canonical CG and UA base pairs, but we also, specifically for RNA, we consider so-called wobble GU base pairs, which are also kind of look like a canonical base pair, but differs a bit from it. But wobble base pairs are very frequent in RNA, not that frequent as canonical base pairs, but they are normally also considered as a part of secondary structure. So we have 3 types of base pairs, and the set of base pairs of these 3 types of base pairs on top of RNA sequence is what we call RNA secondary structure. Now, we have 3 popular ways to represent a secondary structure. One is a planar graph. This is a planar graph. So here, like covalent bonds are shown with the short edges. Base pairs are also shown with the short edges, and these like ladders of consecutive base pairs we call stems. So this is a secondary structure of a transfer RNA or tRNA, and it has 4 stems. Then we have arc diagrams where we stretch the sequence from 5' to 3' from left to right. So this is the same sequence as here. We show it in a single line, and then we depict base pairs as arcs, and as a result we have arc diagram of the same RNA. We also have 4 stems here, so this is the first stem, the outer stem, which is this one, and then we have these 3 shown as arc sets, 3 arc sets. Now, and if like these 2 representations are not computer friendly, machine friendly, the more computer friendly representation is a dot bracket diagram or dot bracket notation, where we also stretch a sequence from left to right, and then we depict base pairs as pairs of matching parentheses or brackets of different types. So this is the same transfer RNA and the same 4 stems. So here we have brackets. So this 2 fragments of opening and closing brackets, parentheses, sorry, is 1 stem, 2nd stem, 3rd stem, and this is outer stem. And here we don't have any unpaired residues between them, so this we have a single stretch of closing brackets or parentheses. Yeah, so we normally decompose secondary structure into elements or building blocks, and for secondary structure we have 2 types of building blocks. We have stems and we have loops. Stems are like so-called stacked sets of consecutive base pairs. So like a set of consecutive base pairs is a single stem, and a single step of 2 base pairs, normally called stacking, because in 3D they are stacked on top of each other, the base pairs, and a single pair of base pairs is called a single stack. So this stem includes 2 stacks, this stem includes 3 stacks, 2 stacks, 2 stacks, 2 stacks. Now, and apart from stems, we have loops. Loops are stretches of unpaired residues confined by 1 or more stems. So this loop, it's a single stretch of unpaired residues, single strand of unpaired residues confined by a single stem, and these loops are called hairpin loop. So this is a hairpin loop, this is a hairpin loop. Often a loop with its neighboring stem, a hairpin loop with its neighboring stem together are called a stem loop. So this is a stem loop, this is another stem loop. Then a loop confined by 2 stems with 2 single-stranded fragments is called internal loop. In case, like a special case of an internal loop where one of the strands is empty is called a bulge loop. And when we have a loop confined by more than 2 stems, at least 3, we call it multi-branch loop or multiple loop or multiple junction or just junction. And for loops that include dangling ends, so this is like beginning dangling end, this is ending dangling end, so this open single-stranded fragments not confined by any stems, So these loops that include dangling ends are called external loops. So in this case, the entire external loop or exterior loop. In this case, the entire exterior loop includes just two dangling ends. In some cases, it would include some intermediate single-stranded fragment. If it would be like two consecutive, for example, stems, then the fragment between them would be part of external loop. Now, before that, I was showing you only the canonical, like the standard secondary structures or nested secondary structures, which don't include pseudoknots. Pseudoknot is a special element where we have like intersections between between stems on arc diagrams. So this is an example of like simplified arc diagrams where a single stem is shown by a single arc, and when we have, for example, this architecture of two stems, we call it a pseudoknot, and this is a pseudoknot of H type. This is a pseudoknot of kissing loop type. On a planar graph, it looks like this. So this is H type pseudoknot, where we like first have a like a fragment of a first stem, then a fragment of second stem, third stem, the second fragment of the first stem, and the second fragment of the second stem. So when we have this arrangement of two stems, this is called H type pseudoknot. When we have this arrangement, like two hairpin loops forming a stem between each other, we call it a kissing hairpin pseudoknot or a K-type pseudoknot. This is how the arc diagram of the pseudoknot looks like. And pseudoknots are special types of secondary structure elements. They complicate the RNA secondary structure prediction by a lot, but luckily in real biologically relevant RNAs, we rarely have pseudoknots more complex than these two types. So mostly we have just H-type or kissing loops and not more complex, because you can imagine more complex pseudoknots with like more intersections, more overlapping orders. It would be, so there are actually like papers, mathematical papers, that try to classify all different types of complicated pseudoknots, but luckily from the biology perspective we have only these two types predominantly. Now in case of pseudoknots, we need more than one type of brackets for non-ambiguous representation of secondary structure in dot bracket notation. So in case we don't have any pseudoknots, we just have parentheses, and it's all easy. In case we do have pseudoknots, we need to introduce additional types of brackets, because if we would have all brackets here as parentheses, then these two opening parentheses would correspond to these two closing ones. But we need actually to show that these two closing ones should correspond to these two opening ones. And for that, these two should be square brackets, and these two should be closing square brackets, respectively. So normally people use parentheses as the first layer of base pairs, square brackets as the second layer of brackets, curly braces as a third layer, and angular brackets as a fourth layer. If we have, for some reason, more than 4 layers, then we start to use Latin letters, pairs of Latin letters, so uppercase as an opening, lowercase as a closing. And this is more or less the most common notation for storing secondary structure. Commonly, if you store it in a separate file, the file extensions would be dot, bracket, or dbn. dbn, I think, is the most common one. Often people just store dbn along sequences in FASTA format. It's also acceptable. And then there is this like fancy dot bracket notation, which is called WUSS, so Washington University Secondary Structure. Here, the types of, it's applicable only for non-pseudonoted structures. And here, different types of brackets and unpaired positions correspond to different types of loops. So like for hairpins, we have angular brackets and underscore signs. For internal loops, we have dashes and parentheses. For multi-branch loops, we have commas and also parentheses. And for dangling ends, we have colons. So this is not not frequently used format, but at least in Rfam database, it is accepted as a main format. Apart from this, we have also some tabular notations. So this is so-called CT file format, connectivity table, where we have each line per position. We have nucleotide, ordinal number, zero-based indexes, and this is two-based indices, and this is the indices of the partner. And for unpaired residues we have zeros here. For paired residues we have indices of partners, and here we have like natural numbering. This is for cases where we have, for example, deletions. So we have like after residue 10 we have residue 20, then in indices we have 10s and 20, and this is natural numbering, just ordering all the residues present in this file. Another variant is BPSec file format, which is like more simplified version of CT. We have just indices, residues, and indices of partners, of paired partners. Again, zeros for unpaired positions, and we have several converters that convert across like BPSec, CT, and dot bracket notations, which is really a regular task to do. In terms of visualizations, we have, as I know, like just one tool that draws arc diagrams. It's R-based tool called Archie. This is its online version, the screenshot for online version. We have more tools that draw plane graphs. So one, like the most popular one is Varna. This is how it looks like. It's Java-based tool. But unfortunately, in case of pseudoknots, it draws some stems like this, because it's like was designed for nested structures mostly. We have some like pseudoknot specific tools like pseudoviewer that finds layouts to show all the stems as actually ladders of consecutive base pairs and not like this. This is less often used tool, I believe. So Varna is the standard. Unfortunately, Varna is a Java based, so it's not incorporatable into web servers, web solutions. But luckily, recently, Denis Incarnati resolved this by developing RFU.js, which is basically as a JavaScript version of algorithm drawing secondary structure as it was in Varna. So basically, it's Varna, like web compatible Varna, but not only. And also, we have specific tool to draw secondary structure for sequence alignments to highlight conserved base pairs and conserved positions. So this is R2R tool by Sasha Weinberg, which is also incorporated in a number of prediction tools. And I think it's this visualization is also a part of RFU.js now. Here, like in green, we have highlighted the conserved base pairs. In normal letters, we have like conserved positions, conserved residues. With dots, we have positions that are conserved but with different base types. And like the color red or black define like how conserved it is. Like 90% is I think red and 70% is black or something like this. And yeah, so the most important is highlighting conserved base pairs with green color. So this is alignment specific. This is web compatible. I remember this. Another important tool is R2DT. The kill feature of R2DT is that it has for a number of templates, for a number of known non-coding RNA families like transfer RNAs, ribosomal RNAs, but actually all the families of Rfam and some more, it has standard layouts, and by sequence input, it actually draws secondary structure diagram in a standard layout for your specific sequence. Why it is important? For example, because like if you have a ribosomal RNA, which is like thousands nucleotides long, all the different tools, automatic drawing tools, would present secondary structure in a different way, but it would be not interchangeable for biologists. So for biologists, we need to use a single like layout for all the ribosomal RNAs to actually identify easily where is which domain, where is which stem, where is which loop. So all the different sequences are represented in the same way. And R2DT does this, exactly this, for known non-coding RNAs, which is very helpful for some cases. And databases. So we have a CRV database, which is a very old database, the oldest database, I think. of alignments of non-coding RNAs, and these are, as I know it, human curated alignments. It has not many, so I think like 100 or 200 alignments, I believe. Now we have the most popular one is Rfam database. Now it includes more than 4,000 families, so I think it's now version 15.1 even. So it has more than 4,000 families. For each family it has an alignment, not necessarily human curated, possibly automated one, and for each alignment it has a consensus secondary structure, again not necessarily curated, maybe predicted by alignment, but yeah, so this is a rich source of non-coding RNAs, short non-coding RNAs. Yeah. Now, in terms of data sets for like training and testing deep learning models, one of the most common is BPRNA1M, which is basically a compilation of CRV, Rfam, and some minor databases. Yeah, so like the drawback of this dataset is that actually the same as drawback for using Rfam, and so why it is used, so it's secondary structure, right? So it's used for training and testing deep learning models for predicting secondary structure. So it's not entirely suitable for predicting secondary structure from single sequences, because in Rfam we have actually like conserved secondary structure predicted for an alignment, which is a bit different from single sequence specific secondary structures. So a given sequence may have non-conserved part of secondary structure, and that we actually want to predict, right? So we should be careful with what data you use for secondary structure prediction, because Rfam and CRV include alignment-specific secondary structures, and the only like the only data that you can be 100% confident of is the PDB derived data, because which is a minor minor fraction of this data set, and only in PDB we derive secondary structure information from known experimentally derived 3D structures. So we experimentally determined a 3D structure, and then we annotated what secondary structure it represents. So we should believe only in these secondary structures, and all the others are just our how the world is not perfect. Now we also have a number of databases which store like 3D fragments of RNA that correspond to different loops of secondary structure. So for each experimentally determined 3D structure, we annotated secondary structure and decompose 3D fragments into stems and loops, and then we catalog and classify those loops and store them in different ways. So we have Fra-based database, which stores stems, loops, and some dinucleotide steps, some multiplets, base pairs, and single residues. We have RNAcosmos search, where you can search for loops of specific sizes. So you query a loop of specific size, and you get all the 3D fragments that correspond to these types of loops. We have RNA 3D motif atlas, which also have hairpin loops, internal loops, 3-way junctions, and some higher order junctions. They store not the loops themselves, but the interacting residues of those loops classified into motifs. And also we have RNA bricks, which actually does the same thing, but also have this coarse-grained representation where each dot is a loop and stem, and we have representations for each 3D structure based on this stem loop decomposition. And we have many more databases. These are the most popular, I believe, or that I'm more aware of.